Now, the organization of gray and white matter in the spinal cord is again very important, and we must know how this gray and white matter is organized when we look at the cut section of the spinal cord. Now, there is one basic difference when we talk about or compare this gray and white matter organization in the spinal cord from the organization in the cerebral cortices is that the gray matter is located centrally in the spinal cord, whereas in the cerebral hemispheres, it is located more peripherally. So this is the basic difference when we talk about or when we look at a cut section of the spinal cord. Now over here, when we just look at or how we divide, for obviously the purpose of study, we divide white matter into a posterior funiculus, and then we have an anterior funiculus which is more anteriorly located. They are again the bundles of tracts that are being located in this posterior anterior funiculi, and then we have a lateral funiculus. So depending upon the location, either anteriorly, posterior, or laterally, we have these three funiculi in the white matter. On the other hand, we have the gray matter, which is being located or which is being organized rather in a butterfly-shaped manner, and it is more centrally located in a spinal cord. So namely, we have this posterior median sulcus, which will help us identify the posterior gray horn of the gray matter. This is the gray commissure, which is basically a communication between the two horns of the gray matter. Then we have the posterior horn of the gray matter, the lateral horn, and namely the anterior horn of the gray matter out there. Now, these are the three horns of the gray matter. Majorly, the cellular bodies, the nerve cell bodies are residing in these particular horns out there. So the neuronal groups can be divided into a nerve cell group in the anterior gray column, which is further divided into a central medial group. The medial group is further divided into posteromedial and anteromedial sides. And then we have a lateral gray column, which is not subdivided. So these are the major subdivisions in the anterior gray column: central, medial, and then we have the lateral subdivision. Finally, we have the nerve cell groups in the posterior gray column. The posterior gray column, basically there is residing the substantia gelatinosa, nucleus dorsalis is present over there, and then we have the nucleus proprius. The visceral afferent nuclei are also residing inside this particular posterior gray column or the gray horn that is present over there. The nerve cell groups in the lateral gray column are also present, and they're not specified out there or they're not subdivided because there is a smaller column as compared to the anterior and the posterior gray columns out there. So this is the general organization when we divide the cellular groups into the gray horns, that is the anterior gray horn and the posterior gray horn mainly. This is a diagrammatic representation. When we look at the cut section of the spinal cord in a micrograph, it looks like this particular, and this is basically the dorsal root. This is the ventral root that we were talking about, and this is the ventral horn, and this is the ventral root as compared. This is the central canal, which is placed centrally, and the CSF is basically bathing inside this central canal. So this H-shaped gray matter under the micrograph is basically characteristic for identifying the spinal cord. This is the dorsal horn that we were talking about. Now, a more schematic representation of how the gray matter is organized is basically, this is how the sensory nuclei are placed. We have a somatic sensory nuclei, and then we have the visceral sensory nuclei that are basically innervating the different organs. Then we have the motor nuclei that are again subdivided into somatic and the visceral nuclei, and this is just in particular with the gray matter. This is again a micrograph which is depicting how it looks. The gray matter, the anterior horn, we can identify the central canal, the gray commissures, the anterior gray commissure, the posterior gray commissure, and the anterior gray horn can be even visualized over here. So this is the posterior horn, we have the lateral horn, and then we have the anterior gray horn, which can be identified in a micrograph. So when we look into the white matter, it is basically divided into these three squads. That is the mainly we have the posterior funiculus, the lateral funiculus, and the anterior funiculus, and there are basically tracts residing inside these funiculi, inside these bundles basically, and they're basically making the roads towards the higher centers of the brain, and obviously taking back the messages towards the effector organs as well. So here we went through the general organization of the spinal cord, how we have the cellular bodies as well as the tracts arranged in a particular manner, and how the spinal cord basically corresponds towards the vertebral column, and then the spinal nerves are formed and they exit the spinal cord towards the effector organs. That is being summarized and discussed over here. So that is the end, and thank you for watching sqadia.com.